My name is Lance Collins. I'm the Joseph Silbert Dean of the College of Engineering, and it's my great pleasure to welcome you to what is going to be a fantastic celebration of one of my favorite people in the college, if not the world, Mike Shuler. So Mike has been an amazing leader. You're going to hear a lot about all of the things, his contributions, his scientific contributions that he's made, and many of you made them with him. That's not something that I can say much about. So I'm going to really sort of focus on what he's done as a leader within the college. He is officially going to be stepping into emeritus status starting July 1st, and we all felt like this was really a good time to recognize all of those contributions that he's made. We have a fantastic agenda today, as you all know, and it's sort of kicked off with my friend George Georgiou, who is the Laura Jennings Turner Chair in Engineering at UT Austin. So we'll be hearing a lot about those scientific contributions, but as I said, I'm going to really be focused on Mike's various terms as a leader in the college. So Mike came to Cornell in 1974, and what was then the School of Chemical Engineering. And then later, between 1998 and 2002, was the director of that school. Now, the seeds of bioengineering had been sown in chemical engineering decades earlier, but Mike was the one that really brought it into prominence. And as the effort grew within the school, he was a key element in adding the important term biomolecular to the title of the school, really signifying a fairly substantial shift that was going on chemical engineering worldwide, but in some sense, importantly, at Cornell. Now, Mike has led many other efforts to integrate life sciences into engineering, and I would argue, importantly, engineering into the life sciences. He held and formalized the university's first bioengineering program in 1994, and then he played some stewardship roles throughout his term at Cornell. But it was during Mike's tenure as the Chemical and Biomolecular Engineering Director that the groundwork for Biomedical Engineering Department was also being laid. In 1997, the graduate field of biomedical engineering was founded, and it's spanning 5 colleges and 11 departments. The Biomedical Engineering Advisory Board was created, which Mike was a part of, and was instrumental in bringing really prominent people here to help guide the early development of the department. And then Mike was the one that was instrumental in getting the department sort of its official status. And then he was the founding director and held that position until 2014 when Mary Lane Vandermeulen stepped into that role. So by the time we had launched, there were maybe as many as 100 different programs across the country. So hiring was really fiercely competitive. And so it was incredible that Mike was able to attract really a stellar faculty to biomedical engineering. And it was really the power of his sort of personal touch, his personality that was so attractive to people that were under consideration. And many have spoken in eloquent ways about the role that he's played as a mentor to them all in terms of developing their abilities to be great leaders, academic leaders within the field, and simultaneously, Mike shouldered all of the administrative burdens to free them so that they could be successful. So Mike created an amazing momentum, and then, as I mentioned earlier, that Mary Lane Vandermeulen has stepped in to continue that growth and development in a fantastic way, and notably, 3 years ago, the department officially became the Nancy E. and Peter C. Meinig School in recognition of a transformational gift that was made. Now, just to put this into some context, the first named department in the college is the Sibley School of Mechanical and Aerospace Engineering that was named in 1885. So it had been well over 100 years since that had happened, and we got our second named, and then not long after that, our third named department, which was Chemical and Biomolecular Engineering. So just to put this into some context, what started as a small group has now risen to prominence in the sense that, U.S. News and World Report ranking places our biomedical engineering at 14, which, given its relatively recent start, is pretty remarkable. And a lot of that development and growth we attribute to Mike. In addition, I just wanted to add a more personal touch. Mike's played an instrumental role in my own career in a couple of ways. He's been a mentor, and at one point, I was chair of mechanical and aerospace engineering at the same time that Mike was chairing biomedical engineering. And there was a kind of important juncture that we had reached. And there was a important biomechanics group that had been built. Don Bartel was the founder of that effort, and Don had reached the point where he retired. We had a retention loss roughly at that time, and there was this critical moment about the future of the biomechanics effort within mechanical and aerospace engineering. And I needed help with that. I needed somebody to provide me with some guidance. So I went to Mike, and I sat in the office, and the style that Mike has, he was fumbling with a cup of tea and a tea bag that was not being cooperative and so forth. And so there was this kind of very casual conversation in which he so clearly laid out why we needed to rebuild biomechanics. That simple conversation, probably less than an hour, remains in my talking points as dean to this very day. What he said, and I think it was the right argument, was that if we can continue to have all of the earlier, older disciplines in this effort, over time, that's going to pay dividends. It's going to take longer to build, but it's just going to be stronger when we get there. And Mike was so instrumental in that. It was a quiet, in his usual very quiet way, providing me incredibly important guidance. So then there was one final time where I went to Mike for advice, and that was, in fact, when we were looking for the successor dean. Kent Fox had stepped down as dean, and there was a search. Originally, it was an external search, and then the decision was made by the search committee to look internally for a dean candidate. And the first name I heard come up was Mike Shuler. And I said, okay, fine, great, we're in great shape. And that was, to me, the beginning and the end of it. And once again, this time Mike reached out to me. We reversed roles, and he encouraged me to really consider becoming the dean. It was not something I had planned, but again, in that sort of quiet way, we had an important conversation, which brought, in some sense, built some of the confidence I would need in order to step into that role. And it's really just incredible looking back on that, how important that was. Now I'm completing my 8th year, so I guess something about it and me have worked. Let's just leave it there. So I just think it's just fantastic. It's wonderful to see you all here to join in on this celebration of one of, I think, Cornell's most important leaders. Mike, I just wanted to relay a couple of things. So one, I think you received a letter from Kent Fox, who was the dean that worked closely with Mike. Then he became provost at Cornell, and now he's the president of the University of Florida. He was unable to attend, but he sent you a letter, and he identified many of the same qualities that I've already spoken to, so I won't read the letter, but it's a beautiful letter, and it was his way of recognizing you and his apologies for not being able to attend. So with all of that, I wish you all a fantastic day and a celebration. I can already, it's part reunion. I can see there's already people that are conversing and probably haven't seen each other in a few years, so that's fantastic. And I just love this idea that we're all here to celebrate, as I said, one of my favorite people. So thank you. Thank you for allowing me to say a few words. So you're going to see something. I'm going to end my comments, and then I'm going to be immediately kind of walking out. But Mike, it's not because of you. It turns out that my daughter graduated yesterday from high school, and I've got a bunch of family in the house, so I got to go and attend to them. So I'm going to be running off. But I really wish you all a great day. So let me introduce our next speakers, who are Abe Struck, who's the Director of Chemical and Biomolecular Engineering, and Mary Elaine Vandermeulen, who's the Director of Biomedical Engineering, to say a few words. Thanks.
So very good. So my name is Abe Struck. I'm the William Huey Director of the Robert Frederick Smith School of Chemical and Biomolecular Engineering. So for many of you in the audience, our school's name has at least doubled in length since you were with us in Olin Hall. I'm going to speak briefly about Mike's research, a view that I have a distant perspective on that allows me to make simplifications and approximations that you can correct during the day. We're going to hear from the experts who trained under Mike and know this story better. My goal is to make one point about my perspective is that Mike thinks differently about research problems than almost anybody, than engineers do, than biologists do, than medical doctors do. And I would even go a little further, and this is one of the things I'd like to gain some insights in from all those of you who trained with Mike and were part of this trajectory, is that honestly, if you didn't know better, at any given moment in Mike's career, you might think he was a little out of whack in how he was defining his problems and pursuing them. And if it weren't for the incredible track record he's had, you could have questions about his sanity. So just just very briefly, in the earliest days when both biologists and engineers assumed that a living cell was an intractable mess, Mike set about to write down the equations that define its dynamics, its internal dynamics of the central biochemical processes. And he used computers that were less powerful than a handheld calculator of today to take a fully chemical kinetics engineering approach to understanding metabolism and signaling and gene regulation. He was doing systems biology 20 years before that term existed. And in many ways, he was laying the quantitative foundation that has been the basis for how we define biological engineering ever since. In the middle of his career, as industry and academics were trying to converge on just a few cell types to work to be the workhorses of biomanufacturing, Mike was often left field working with insect cells and plant cells, a place that very few people were going to follow him until he showed that this is an extremely powerful set of platforms and have now had major impact in industry, including being the basis for producing natural products like chemotherapeutics that really had no other route. Starting in the 90s, Mike seemed to be really nuts. He set about to make physical models to represent mathematical models of the real human body. And this sounds like the plan of a schizophrenic. I think in some sense that Mike is. He's someone who has a deep appreciation for the mathematics that underpins engineering analysis, and also a strong commitment to practical realization of a system and experimental outcomes. So no one at that time, I think, could see where Mike was going. And indeed, no one paid much attention for, I'd say, about 15 years. But by then, Mike had developed a whole new perspective and set of approaches. on tissue engineering, on drug discovery and toxicology, with his so-called body on a chip. And today, in these last about 8 to 10 years, this is now an entire new field of bioengineering. With almost journals filled with people working on these ideas that Mike had pioneered. So even with hindsight, I still have to say that I think Mike is a bit loony, but seems to be loony in just the right way. And so we're lucky today to have many experts who were co-conspirators in 45 years of an attack on conventional ways of approaching research. And I'm looking forward to gaining a few new insights into the methods behind the madness. So thank you. Enjoy the day, and Marilyn will speak a few words about leadership.
Let me add my welcome to everybody in the room as well. So I am the James M. and Marcia McCormick Director of the Nancy E. and Peter C. Meinig School of Biomedical Engineering. And as Lance pointed out, the naming of biomedical engineering is a landmark not only for biomedical engineering, but for the College of Engineering, given that our previous school was named when the university was founded. But that naming came about because of discussions that Mike had many, many years ago with Pete Meinig. And Pete Meinig, for those of you who don't know, was the chair of the Board of Trustees here at Cornell. And so it was really, the naming was a final buy-in on the vision that Mike had for this department. So his contributions, as Abe just explained in research, are many and have led to many recognitions, including the National Academy of Engineering at a very young age. But his contributions to his vision and leadership in the College of Engineering are really unlike many others. There's few people who have chaired two departments that have grown into what they are today. Like I said, they're both named, they're both growth areas. And so really for many years, Mike Shuler was, and I'd say still is, synonymous with biomedical engineering at Cornell. And in fact, he really was biomedical engineering at Cornell. If you said BME, it automatically brought to mind Mike Shuler. The successful establishment of the department in 2004, it wasn't just his vision, but it was also his perseverance and his hard work. And I think I actually saw one letter, an external letter for, describing it as, Mike banged his head on the wall of the Cornell administration for many, many years and did so relentlessly, but because he had this vision, and therefore that's why he was successful. And so he really, from the implementation through the hiring, through where we are today, took an amazing amount of effort, and it's something I think few individuals would have undertaken, and many, a year or two into this multi-decade process, really would have walked away. What's really unique, I think, is that the Department of Biomedical Engineering was formed with a very strong vision and a core set of values. And all the hires under Mike bought into that set of values, and that created a very shared experience and a unity that's really unique and strong. As his successor, I can say that he cast a shadow that's long and that's large, but it's not dark. And it's interesting, when I started, several faculty came up to me and said, wow, I would never want to follow Mike as chair. Look at what he did. You're sunk from the beginning, sort of, like you don't stand a chance. But I think that's a phenomenal thing. And actually, 4 years into this job now, I understand that even more than ever. When I started, it was intimidating, but now I get it, having done this position and the decisions he made and again, the core vision that he had. And in fact, my contributions to this special issue are entitled, What Would Mike Do? Because that's something I ask myself very frequently because he did it so well. Mike, biomedical engineering is going to miss you. We already miss you as chair. We're going to miss you as a daily colleague, but we're not saying goodbye. As a matter of fact, if you think it's goodbye, we don't think it's goodbye, just for the record. And it's not farewell, but we look forward to having your lab continue and the contributions you'll continue to make, although less frequently. We don't you're not going to be in the classroom, and we don't expect you at faculty meetings, but we do expect that we'll still see you, and we look forward to that. But I also think you've really earned your fishing trips, and so please keep that in mind. And now it's the fish's turn respect Mike. So again, thank you all for being here. And I have the pleasure now of introducing Matt DeLisa, who's going to kick off our first session with this invited talk. Keynote.
Okay, good morning and welcome everybody, and thank you for joining us here today on this really tremendous occasion. I have the great pleasure of introducing our first speaker this morning, our keynote speaker, George Georgiou. George is currently a professor at UT Austin, and he received his bachelor's degree from the University of Manchester in the UK, and then his PhD here at Cornell, and that was under the careful, watchful, and somewhat loony eye, as we learned, of Mike Shuler. His current research program is focused on understanding the serological antibody repertoire in human health and disease, and on the discovery and preclinical development of enzyme and antibody therapeutics for cancer and for inborn errors of metabolism. George was elected to the National Academy of Engineering in 2005, to the National Academy of Medicine in 2011, and to the American Academy of Arts and Sciences in 2016. So I have a whole half page here of other accolades, and I was actually prepared to read it here, and instead I'm going to fold this up and put it away. And I think in the moment here where we are honoring Mike for many things, many accomplishments, but more importantly, and I think for many of you why you're even here today, for mentorship and guidance that he provided, I do want to say a few words along those lines. Last night I had a chance to thank Mike directly and share my gratitude for everything that he did in helping me get started in my very early career here at Cornell. In that spirit, and with two very important individuals for me here today in the audience, I'd like to say thank you to my Mike Shulers, Bill Bentley, who's going to speak a little bit later this morning, and also George. Bill was my PhD advisor, and George was my postdoc advisor, and clearly without those two individuals helping me along the way, keeping me focused and on track, I certainly wouldn't be here today. And so in the spirit of recognizing phenomenal mentors, let me please welcome George Georgiou.
Thank you, Matt, and thank the organizers for this great honor to say a few words in celebration and honor of Mike, a person who had an immense influence on my life. I also like to express my angst for having to talk for an hour, and in light of all the other speakers that will be giving a talk about Mike. So how do you actually talk for a man for an hour, about a man for an hour, and then be followed by another group of speakers that will also say great things about his various contributions? So I thought instead of doing that, actually, since Cornell is a very intellectual place that's very dear to my heart, give a more philosophical talk on serendipity, models, time, and original sin. I don't think I left too many things out. But I think it's all about celebrating Michael Shuler in various ways. So actually, all these topics do connect to Mike in some way. The original sin, probably not so much. But first, let me start with a disclaimer, and I'd like to manage expectations. In the great man's own words, okay, I'm not exactly the best speaker. I really believe that. It's taken me a lifetime to perfect my presentation skills. And this is an excerpt from my recommendation letter to prove the point for a faculty position, which I unearthed in some dungeon at UT Austin and had been very helpful in terms of providing some material. So his oral presentations are quite adequate, but need a little bit more polish. And they still do, Mike, so apologies for what's going to follow. In fact, apart from being true, it's also been a great phrase to use in recommendations for other trainees, with the notable exception of Professor DeLisa, who was born a good speaker. So, every story starts with a prologue, or should start with a prologue. And one of the most famous phrases in molecular biology and biology in general is the first sentence of the double helix by John Watson, who said that he's never seen Francis Crick in a modest mood. And if you ever had the pleasure to meet Francis Crick in person, I only met him once, he was a bigger than life personality who'd made his presence known and also had a little bit of arrogance. However, this is the exact opposite of what Mike is all about. He's always been the modest, most humble person I've ever known, and also he's always been known, and everybody can attest to his equanimity through situations that could have made other people more upset. But let me begin. Serendipity. So somehow I ended up at Cornell. A few of you know the story of how I ended up at Cornell. It was because of really bad geography and love. Love relates to a girl that was at Columbia. Bad geography relates to the fact that I didn't appreciate that Ithaca is actually not New York City. It is a true story. And I came to visit said girl during my spring break, and we had a wonderful time in New York City. We didn't get killed because at that time, New York was very, very dangerous, early 80s, and we're walking around Harlem. And then I visited Cornell for an interview, and I thought, oh my God, this place is really in a different continent by European standards. But this is where I decided I'm going to go, and here I am at Olin Hall in 1981, with much more hair than I have now, in the office that I occupied for 5 years. So of course, as you know, once you enter graduate school, you have to actually select an advisor. And I had to select an advisor, and I didn't know how to go about the process. And I thought and thought very hard, sought all the information I could, and of course I made a decision the right way, okay? I tossed a coin. This is also a true story, because I was thinking of working with Professor Gubbins in this amazing area of basically pressure, volume, temperature, three-dimensional representations of phase diagrams, which to me sounded so exciting, but also thinking about biotechnology, and couldn't decide. I tossed a coin. Well, actually, I lost, because had I won, whatever it is, I would have worked with Professor Gubbins. So I decided to work with Dr. Shuler, and I had no idea what to expect. And here starts a long relationship with this man who, at that time, looked like this. He still actually hasn't changed that much. Well, it was actually a good run scientifically. I learned an awful lot, and we'll discuss that in a minute. And 5 years later, time came for me to look for a job. And I was thinking, first of all, I cannot cut it in academia, okay? Secondly, why would I want to go to academia? I mean, it appealed to me in some ways. However, it's a daunting career path if you see your advisor and you see how many things he has to do. But Mike really urged me and encouraged me strongly to consider an academic route. So I have to also tell you, my level of self-confidence wasn't really all that great. I was kind of dyslexic, and I wasn't diagnosed until many years later, in fact. And also, I wasn't the best student in graduate school, not because I couldn't master the material, I think, but because I didn't really care. So I did very well in Dr. Olbricht's class, but some of the other classes I found rather, how should I say this, not to my liking. So I ended up graduating or finishing my graduate degree with the amazing GPA of 3.2, which in graduate school is actually pretty darn miserable. So that was another reason why I thought, well, how am I going to be hired, or why should I spend my money putting stamps on envelopes? There was a time when you had to do that, actually, to send applications out and type applications to various universities, which is going to be a waste of time. But Mike said, no, you really should go for that. And I said, okay, where should I apply? He said, oh, MIT, Berkeley, Stanford. And I thought, ha, 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 ha. I don't know why on earth he is saying these things to me. Is he really? Either I managed to fool him or there is something else going on. So I did not apply to those schools because I didn't think that they were in my league, and I applied to some other schools. And one day, Mike called me in his office and said, you should apply to Texas. I said, say what? Texas. I said, Mike, no, I just bought skis. And my wife bought skis too. Said, no, you should apply to Texas. They have a lot of money, it's a good place. I said, okay, we'll apply to Texas. More about that in a minute. But in any event, so Mike wrote a great recommendation letter for me, which I credit for getting some traction with several departments that invited me for an interview and eventually made me an offer. And of course, in his letter, he had to justify the fact that my course performance, although quite satisfactory, it wasn't, is not what we typically expect in a high-quality faculty prospect. Well, because some schools asked for my transcripts, darn it. However, he, in a very elegant way, tried to justify why this is not an issue, and it shouldn't be an issue in my hiring. He said, factors which may have been responsible include difficulty adapting from the English system with a single exam, a girl from New York City. That's a good reason why one may not do well in their classes, and initially some immaturity, a lot of that. These are now not problems. And the reason is because he has adapted, and now he's married to a woman of dual US-Greek citizenship. And here comes the best part, and considerably more mature. All of which is actually true. And this woman, who basically was way, way out of my league then, and still is here in the audience. It's Mary Jean Torres. You can tell why she was way out of my league, it's clear. And she's really the most interesting woman in the world. Not only is she my wife, and has been my wife for the last 34 years, but we got married at Cornell, at Toganok Falls, but also, she's been my business partner when we venture out to biotech. She's an attorney and has done all the initial corporate work for ventures that we started together. So she was my other very strong supporter who also believed in me. I definitely did not believe in me. But Mike and my wife, for reasons that mystified me, believed in me. I applied for faculty jobs, and I, at Mike's urging, applied to Texas, and to my surprise, they wanted to come for an interview. And I remember the day. It was February 10th, 1986. The temperature in Ithaca was 8 degrees, and there was a blizzard. And I arrived in Austin, and it was 70 degrees, and people were in short sleeves. To which my wife, who had also grown up in the Southwest, after about 10 minutes, said, "We're coming here." And I thought, oh, wait, what do you mean? Oh, we're coming here. Truth be told, this is what happened. We agreed to give it a try for 2 years. For those of you who know Austin, it's a blue dot in a red sea, but at least there is some good music. It's now become a very hipster place. It didn't used to be a hipster place back in the old days. It was a little more of a sleepy college town, but it was a good university. Not exactly from a temperamental place, what I would have expected, but actually it was great for us and our family and our career. So, to my surprise, and actually I followed again on Mike's footsteps, I've been at Texas for 32 years, and this is important because just like Mike, I had considered going to different places. When I was a graduate student, Mike had several offers, and the graduate students actually had taken a poll as to which offer he's going to accept. He's going to Wisconsin, he's going to go to Penn. There were a few other places that were recruiting him because he was, and he continues to be, a superstar. And he decided to stay at the same place. And for a number of reasons, I decided to stay in Austin all these years and built my family and my career there. So, chapter 2, models. Now, if you worked with Mike in those days, and I think throughout your career, at some point, you will have to work on a mathematical model. And actually, I was proficient in math and decent in computers, with computers. Decent means if proficiency with Fortran 4 qualifies as being decent, it doesn't anymore. But my first project was actually to build a model of an organism, a fungus, growing on a solid substrate. And when fungi growing on surfaces like your bread, they actually form interesting patterns. And was there a way to predict those patterns by a mathematical model? So I wrote a few thousand lines of code. It took me forever to debug. It would take 3 days to run on a PDP-11. Those of you who don't know what the PDP-11 is, but it was the state-of-the-art machine back then. It was a great experience, but very frustrating because you also have to debug a lot of code, and my coding skills weren't that great. And one of my memories, which actually still stuns me, is that at times I would go to Mike whenever I was frustrated. And nobody can help you debugging. But Mike offered to actually look at my code and help me. And of course, you cannot go through 2,000 lines of code and help someone with their program. But just the fact that he was there and he was willing to put the time and talk to me about phishing while we were going through the code, it was actually such a strong endorsement and it gave me such a sense of support that helped me go through this. And we published a nice paper after this work. But modeling was really not for me because I realized that while it's very informative, I wanted to be able to do something that's really more hands-on and generates data and information. Molecular biology was coming up, and I thought, I'm going to move from chemical engineering to molecular biology. So I went to Mike, his door was always open, and we had a conversation that, again, changed my life. I said, Mike, I appreciate my master's degree. I got my master's degree, but for my PhD, I'm going to go to molecular biology. I'm going to switch departments. And he said, oh, well, that's what you should do. But do you know what's going to happen next? I said, what do you mean? Well, after you get your PhD, you'll have to do 2 to 4 years of postdoc. I said, what's that? Oh, you're going to be paid as a graduate student, and basically you're going to be like a graduate student, but that's what you have to do to get a job in molecular biology. So I said, wait a minute, I'm a chemical engineer. I can get a job, so I'm going to do my PhD and have to work for a pittance for another 4 years. Is there anything else that can be done? And he said, let me think about it. And he came up with an idea. He had just started a collaboration with David Wilson in the biochemistry department, and he said, well, why don't you go to biochemistry, talk to David, and see what you can do, and maybe you can learn some molecular biology. And therein started my trek from Olin Hall to Wing Hall. A beautiful walk in the summer, not so great in January when you have to carry samples back and forth several times a day, and you don't want to be taking the bus, which is usually late. So I spent basically 4 years, almost 4 years in Wing Hall, essentially being trained in molecular biology while getting my degree with Mike in chemical engineering and reporting to Mike and secondarily to David Wilson, who was another incredible mentor and a very welcoming advisor or co-advisor. But I learned molecular biology at a time when very few chemical engineers actually knew much about molecular biology. And thanks to my good luck, I was one of the few, probably the first chemical engineer to actually run DNA sequencing reactions and do a lot of heavy-duty molecular biology work that wasn't really part of the agenda in those days, which helped me greatly later on. So this work led to an interesting finding, again, serendipity. We were trying to produce a protein, and we couldn't find the freaking protein. And the reason why we couldn't find the protein is because it was getting stuck. It was aggregating. It couldn't behave well in a recombinant cell. That was something that was becoming apparent, but there weren't that many reports. And in fact, the paper that we published with Mike was the second report on this topic. In this electron micrograph, what is shown is areas inside the cell that are different color and different density and reflect the aggregation of a protein inside the cell. So I thought, this is an interesting phenomenon. Maybe this is part of what I should study for my work as an independent faculty member. Why some proteins cannot really fold well in the cell. And I did that, but I have to contrast Mike's optimism and continuous support with another biochemical engineer that I met, a very famous biochemical engineer that I met early on in my career as soon as I landed in Austin. So we invited another major figure in the field at the time who gave a couple of lectures, and I told him what I was planning to do, and he said, young man, you're going to fail. This is a solved problem. Everybody knows how proteins fold, and basically you're going to waste your career. So move on to something else. It was so demoralizing because I felt that, oh, wow, probably I'm embarking on something that nobody cares about. Turns out that he was wrong, and he didn't have the vision and the foresight that Mike did. So one of the things that actually was very, very interesting and important is that Mike not only supported me in getting a job, but he insisted that I apply for a Presidential Young Investigator Award, which at the time was a really big deal because the program had just started. And in fact, he nominated me for one. And I don't know what happened. I had this letter. It was in the dungeons at UT. I found it, and he said that I will do something important, and my work will be at the interface of biology and biochemical engineering. That part is true. For some reason, it worked. And I had no idea why it worked. And in fact, my chairman called me. I was working from home on a proposal, and he told me that I got a Presidential Young Investigator Award in my first year. And I hung up the phone. I told my wife, it's a mistake. They will surely call me back to say, oh, sorry, it wasn't the case. But it was the case, and after a few weeks, I realized that, okay, this is a good start, and then something maybe good can happen from all this. I have some money, I had some grants, so that was what set me to the right course for my career. And through those early years, in fact, on more than one occasion, I sent Mike my papers, and he was kind enough, despite his many, many obligations, to actually read through them and give me comments and continue to be supporting and continue to be a mentor, even after I wasn't really one of the people that he was immediately responsible for because I had grown up. I had flown away. So what comes to mind when I think about Mike? Well, there are numerous properties, numerous qualities that are really unique in combination and individually. Intellectual curiosity, his unbridled optimism. I don't know how he manages to be always optimistic. It's almost irritating. His equanimity, decency, and incredible sense of integrity. These are words that we don't use as much these days, but actually they matter to the world as much now as they always did. And I learned so many lessons. First of all, to believe in your students and go to great lengths to support them. Actually, that was a big burden when I was starting out as a young faculty, because I thought, oh, if I screw up, then the people that are under me will also be affected in a negative way, and didn't want that to happen. mentoring is a great responsibility and a privilege, and it's absolutely the case. It is, and one should take care of the students because ultimately this is what matters. It's the people that matter, not the freaking paper or the proposal. Another thing that I learned from Mike is to explore a new scientific area every 8 to 10 years, which is what he's done over and over and over again in his life and his career, and Abe referred to that, and I've witnessed that. And it's hard because you have to go to a new field and you have to learn that field, and then you have to be connected to the people in that field. It takes a lot of effort, but it keeps you on your toes. So it was actually a good model to emulate. And finally, everything in biology can and should be quantified. It may be hard, but actually it's very important as an engineer and as someone who thinks in holistic systems terms to try to put some quantitation into anything that relates to biological research. And that's part of what our profession is about. Okay, chapter 3, time. So, as I said before, I have a lot of time allotted, so I'm not so sure what I do. Initially, I was thinking, my first idea was, okay, my keynote will be last. If I buy a case of wine, then we can have an early break and everybody will be happy. But unfortunately, my keynote is at 9 a.m., and that would not work very well. So I have to do something else, and probably the only thing I can do, other than be very repetitive and talk about Mike's qualities in a way that will probably be covered by the other speakers later on, is say a few things about what I've done since I left Cornell and how the lessons I learned from Mike have followed me throughout my career. So the first lesson that I mentioned before is exploring a new direction every 8 to 10 years. So we had a good run understanding why proteins fold or do not fold in bacteria in particular, and doing a fair amount of molecular biology work as well as engineering work that ultimately also led to some technologies that I will discuss briefly later on. But then about 8 years later, I started getting involved in a different area, slightly different area, which is engineering and manipulating proteins and developing methodologies to manipulate proteins to engineer molecules with particular properties of interest for therapeutic or other purposes. Initially for other purposes, eventually, as I will discuss later on, for therapeutics. And then, about 8 years later, we become interested in the genetics of the TAT pathway. It's a secretion pathway in bacteria that Dr. DeLisa knows very well, and on RNA degradation. There are two things I wanted to mention in this regarding my own research path. First, I tried to follow Mike's advice, as I mentioned before, but secondly, as we moved from one field to the other, it helped basically take projects that we start in the lab and nucleate in the lab and actually allow those to the trainees that got the faculty positions to take the project with them, which doesn't happen often in biochemical engineering. So because, life is, the world of science is actually quite big. And if you think about it, if you are well-positioned in the field, you can always find something else to do. But for a trainee, it's very important that they continue and expand on something they've been trained on. So Matt, thankfully, continued some of our work on the TAT pathway for a while and had some great contributions, and several other students, like Jennifer Maynard, who's now a professor in chemical engineering at UT Austin, Louis Massip, and several others actually continued in the path that initiated at their PhD. So in addition to publishing papers, actually the work ended up being useful, and some of the manufacturing methods related to protein folding were used for the production of drugs, a couple of, actually 3 approved drugs. The protein engineering technologies were licensed to pharma and biotech for a number of discovery programs. And as part of also these efforts, we engineered an antibody that won approval. This is an antibody that protects against inhalation anthrax, and it's been stockpiled by the Department of Defense. Well, this continued on, but about 10 years ago, as I was getting closer to a milestone birthday, like 49, I decided that I needed to do something else. I wanted to get away from bacteria and have changed my program completely. So after the first 20 years, it was time for even more changes. And that's when we regrouped and retooled and started a program in human immunology and also in protein therapeutics. And the protein therapeutics program in particular did end up leading to a number of experimental therapeutics that are in clinical trial. And it was an eye-opener because it enabled me to enter in a more extensive way the world of biotech and understanding drug development, and both preclinical and clinical, which is a different world. And Anne may talk about that later on. But also, going back to serendipity, part of what catalyzed this is that when I was at Cornell, I learned how to ski. First of all, you'll never learn how to ski well in Greek Peak, okay? Second, you'll never learn how to ski well when you're 22. But nonetheless, I loved skiing. In 2008, however, through some unfortunate events, I broke my tibia in 12 places. And I had to stay in bed for 2.5 months. And this was actually, in some ways, very good for my science because I got bored out of my mind. After a week of watching TV and books, I started really working. So I wrote a couple of proposals, and those proposals, for whatever reason, the Vicodin may have helped, actually got funded and catalyzed the work that we started in something that I had no experience beforehand. So in doing so, we had to set up a different model. Every time you work in a new area, you really have to understand how to structure this effort from a logistical standpoint. Logistic matters. So basically, the lab, in some ways, began to work as a small biotech. So we had biological insights, and we will come up with a therapeutic molecule, a molecule that probably could be therapeutic. Biological insights meaning if we understand something about disease, then we will know that a particular disease agent may be critical. So if you can act on that agent, then you can actually, in some manner, then you can ameliorate the effects of disease. And then you come up with a molecule. You have to prove that this molecule will do something useful in vitro and then in vivo. And then you have to do further work to develop a clinical candidate, a molecule that has the requisite properties to be a possible human therapeutic, which is really, really, really hard. We don't appreciate that in academia, but and sometimes it's not glamorous work, but it's incredibly important. And then you have to do a little bit of bioprocessing, because if you can't produce something, especially with proteins, you don't have anything. And in fact, most interesting drugs are killed by the pharmaceutical industry, not because of other reasons, not because of side effects or bad biology, but because people cannot really make them in a financial way, in a meaningful way, at least not initially. So with all that, if you have that package to some extent in place, then you can go out and try to raise money from investors, and eventually the investors will facilitate the next steps in the process, which is basically getting approval from the Food and Drug Administration to test the drug in clinical trials. So it worked actually quite well. We started doing this about 2009, and as I mentioned before, in earlier studies where we didn't get engaged in the entire process, we had one drug that did make it to approval. But then after that, I founded a company called Aglia, which is now public. It's one of the few biotechs in Austin. which has several programs in clinical development, and a second company which is in Boston, which has a molecule called kinase with a Y. Now, for those of you who are biochemically sophisticated, there's a reason why this is spelled with a Y. A, it's a biochemical reason, and B, because when people in this field think of kinases, they think of kinases with an I. So you have to explain what kinase with a Y is, and it sticks in people's head. But anyway, it's an oncology drug, and actually we will start GMP manufacturing later in the year. Now, founding companies, a lot of people found companies, but what I find to be very important is not being involved only in the formation of a company and setting the stage for the company, but also be continuously involved. And both of these companies have an active relationship with a laboratory. A lot of the work is done with contracts with the university. It's been a great collaboration because we get access to data that we could not possibly have for publications like non-human primate data and even clinical data, number one. Students and postdocs work as consultants or have equity in these companies, and the university, of course, benefits as well from the process because they collect royalties and other milestone payments. The sacrosanct rule that you absolutely have to abide by is to make sure that you understand what constitutes a conflict of interest. Never, ever, ever do work that is proprietary because we're a university. Everything that students and postdocs do has to be in the public domain as it should be, and we publish on everything. So, I have another 15 minutes. So I'll talk about the other topic of our work, which is relating to antibodies. Well, antibodies are fascinating molecules. Of course, all molecules are fascinating, but some molecules are more fascinating than others. Antibodies are fascinating first because, well, right now, antibodies are the fastest growing class of drugs in the biopharmaceutical industry. As a society, we spend about $90 billion a year on antibody drugs, and the market grows at about 15% a year. This is also the reason why the healthcare system may go bankrupt, but let's not go there. These are expensive molecules to produce. They're very powerful, but expensive. But also, apart from their technology and financial weight, antibodies are important because they protect us against disease. If you cannot produce antibodies, you're going to die from an infection. So we all need antibodies for our lives. So antibody immunity was recognized at the end of the last century. And in fact, von Behring, who was an SOB, got the first Nobel Prize in medicine for the development of antibodies, or actually therapeutics to toxin and diphtheria and other diseases that basically involved immunizing horses and taking blood and putting into people so that they can be cured from an infectious disease. If you're interested in this topic, I recommend you watch a Netflix series. It's a German series called Charité about the early days of the development of immunology and antibodies. There are several interesting things that I have to say about the discovery of antibodies. First, von Behring got the first Nobel Prize, even though most of the work and the concepts were developed by Paul Ehrlich, who got the Nobel Prize later. But Paul Ehrlich was Jewish, so it took a few years before it was acceptable for a Jew to get the Nobel Prize. Also, the person who helped in a meaningful way, in a substantial way, he was a Japanese scientist who went to study with Koch, who also was at the same place at the Charité. All these geniuses were at the same place at the same time. Shibasashuro Kitasato, who should have won the Nobel Prize together with von Behring, but he was Japanese. Well, Kitasato, though, was amply rewarded for his efforts and contributions. He went back to Japan, he was a hero, and the emperor asked him, "What do you want?" And he had been in Europe for a few years, and he said, "I want to become a count." There were no counts in Japan, so he was the only Japanese count. So anyway, antibodies protect us against pathogenic agents, and all vaccines, or nearly all vaccines, work by eliciting antibodies that protect you against an infectious agent. How? By recognizing that infectious agent and either directly clearing that virus or the bacteria, or clearing the infected cells. Also, antibodies in many ways protect us against tumor cells as well. And we always have some antibodies that actually are anti-tumor antibodies. We get cancer because we never get sufficient antibodies against tumors because they're not really easy to elicit for a number of reasons. So, Mike taught me that when you want to start on a new project, think about quantitation. And quantitation is really hard because, in this case, because it's medical and clinical research, and clinicians don't think in numbers. This is very important, okay, to keep in mind next time you go to the doctor. So it took a while, and I'm proud of this slide because I had to do a lot of digging to find this information. So how are antibodies made, okay? Well, antibodies are made by B cells. B cells comprise roughly about 2% of the cells in the body. By the way, for those of you, one interesting tidbit is that the immune system actually is an organ. It's a distributed organ because it's not in one place. Your spleen, your bone marrow, your lymph nodes are an organ. But altogether, the size of your immune system, the number of cells of your immune system, make the immune system the second biggest organ in the body after the brain. Well, okay, we don't take into account the skin, but that's because that's a different story because it's also an organ. So anyway, how do antibodies develop? How do we make exquisite antibodies that protect us against disease? Well, B cells are born in the bone marrow, and they're born literally as naive B cells, which means they don't really bind very well to anything. We have a large diversity of different B cells that we generate every day. These B cells circulate for about 6 months, and then they die. And roughly, we have maybe 50 billion naive cells circulating in our body. By the way, if you ask how on earth do I know these numbers, it's because I looked at the literature of people who look at cadavers and also who try to gauge how much radiation to give to patients to kill them or not to kill them, to kill their tumors without killing them. And from extrapolating and a little bit of accounting, you can actually come up with these numbers. When B cells encounter a pathogen, let's say it's an influenza virus or strep or anything, they become trained. And when they become trained, they become what's called memory B cells. Memory B cells produce antibodies that are now specific for a particular agent. They bind with very high affinity and very high selectivity. And some of these antibodies will be biologically relevant. They will help clear the pathogen and protect you from disease. The diversity of the memory B cell repertoire is even greater. We have many more distinct antibodies, but we don't know how many or how many copies of each, and we don't know how many total. Some of the cells that have been trained, some of the B cells that have been trained after being exposed to antigen, mature further, become further differentiated into what's called long-lived plasma cells. They're still B cells, but they're called something else. What do long-lived plasma cells do? Is they produce insane amounts of antibody. How insane is insane? Well, if you take Genentech's best process for the production of antibodies in a bioreactor, something that Ann Lee, our next speaker, contributed to in significant ways, plasma cells actually produce about 3 or 4 times more antibody than what we can still do. And they do that sometimes for decades. They are under exceptional stress because they have to be antibody factories, but somehow they survive. So antibodies reside in the bone marrow and a few in the spleen. The long-lived plasma cells that continuously put out antibodies. And the antibodies now circulate in your blood and circulate in your tissues and survey the entire landscape of your body for pathogens. And if a pathogen is bound by an antibody, then the pathogen will be either blocked from infecting cells or will be cleared. More on quantitation. Humans, normal humans, have between 6 and 17 milligrams per milliliter of antibody in their blood. That's a lot of protein. Basically, to put it in, I love McVitie's chocolate digestive cookies. So to put it in those terms, to replace all my antibodies, I will need the energy in a pack of McVitie's cookies, which is a lot of cookies, okay? So the serum antibody repertoire, what circulates in our blood, has a number of different antibodies, and nobody really had known how many different antibodies we have. When you have an infection, you know that you are infected with strep or with HIV or with whatever else. And how do you know that? You go to the doctor, and the doctor gives you a test, and the test says, aha, you have an antibody response, your body listed antibodies against strep. That's how those tests work. But it doesn't tell you, do you have one antibody or do you have 500 antibodies? How many antibodies are in circulation? So doing a little bit of mass balance, a lot of mass spectrometry, and a few other tricks, we estimate, and we've estimated that on average in humans, we have about 20,000 distinct antibodies in circulation. Sounds like a big number, but these antibodies have to basically do a whole lot of different things. They have to protect us against pathogens. They have to clear junk that is generated all the time from dying cells in your body. They have to also, in some ways, perform an anti-inflammatory role, and we're not going to talk about that. So, but 20,000 different molecular species do a lot of different things. Antibodies, the B cells are born in the bone marrow. They migrate to the blood and the spleen and the lymph nodes. That's where they get trained. And then after they get trained, eventually they become antibody-secreting cells. And the antibody-secreting cells, when they become mature, they go back to the bone marrow and they stay there for a long time, as I mentioned before. And from there, they produce the antibodies that we have in our bodies. Now, when I was in bed, rereading immunology from cover to cover, I realized that we really don't know that much about what kind of antibodies, what are the specific antibodies that are present in circulation. And 100 years after the discovery of antibodies, really, we didn't know whether we have 10 antibodies, 100 antibodies, or 1,000,000 antibodies. Typically, immunologists, when asked them the question, because I first asked my friends, my immunology friends, how many antibodies do we have in blood? They would say, oh, a few million. Well, if you do the mass balance, you say, wait a minute, an antibody has to bind something. If it has to bind something, it has to be present at a concentration of about 1 nanomolar. If you do the math, then the density of blood should be, about maybe 2,000. So the density of water is 1. So obviously, your blood should be, it's impossible to have that many antibodies. So there has to be a smaller number. The reason why people haven't looked at that problem is that it's a technically difficult problem and the technology hasn't been around to address it. The antibodies in circulation change all the time. Antibodies are very similar. We call antibodies immunoglobulins, and therefore IgG is one part of immunoglobulin that we care about. They're difficult to resolve. They're present at very large, different concentrations. And they're produced by cells that are not really accessible. You can't really basically somehow withdraw somebody's bone marrow and then analyze what's there, the cells that produce the antibodies, because that person will be dead by that time. So we developed a technology, which I'm not going to go into detail. It has a lot of moving parts for analyzing the molecular deconvolution, the molecular properties, identifying how much of different antibodies to a particular pathogen or to a particular agent are present in blood. And it consists analyzing both the serum and also the cells and combining that information. It requires a lot of informatics, and it's actually a fairly complicated technology, which is great, because if you have a complicated tool, that means that other people cannot have that tool available to them, so they'll come to you and collaborate with you. And clinicians that normally are a little arrogant, general statement, but often clinicians are somewhat arrogant. Eventually, they come and collaborate because they appreciate the value of bringing something to the table. So what have we learned about the antibody repertoire? Well, this is an example of one of our first studies where we analyzed the antibody repertoire to tetanus. You get vaccinated with tetanus every 10 years. I think it's a great idea that you remember to get revaccinated because tetanus is a pretty bad disease. And if you look at the antibody repertoire to tetanus, this is a particular donor that I care about, me, okay. The first experiment, we had to get a lot of blood, so we couldn't get approval to get a lot of blood, but you can self-experiment. So I went to, I was in Germany at the time, at the Charité, and self-experimented. Now, the German doctors, being very gung-ho, also wanted to draw a lot of my bone marrow, and I asked a couple of my friends, should I do that? I said, you're crazy? They're Germans. They're a little less, more tolerant to pain than Southern Europeans. So I didn't have my bone marrow samples taken at that point. In any event, so each of these bars corresponds to a different antibody, and as you can see, there are relatively few antibodies that are basically conferring protection to tetanus. You basically are protected to tetanus by no more than 20, 30 antibodies that are in circulation, and they're there all the time. They're produced for years and years and years on end. Granted, their amounts decrease, but still there may be sufficient amount to produce, to protect you. And a few antibodies are present at very large levels, very high levels compared to other antibodies. So if you take the 5 top most abundant antibodies, they typically comprise about 70% of all your antibody response to a particular agent. This is true for many other agents. We've analyzed over 100 donors for various diseases, and it's always been the same case. So, and now chapter 4, the original sin. By the way, I'm Greek. We established that, okay, we've been in this country for a long time. But as you probably know, the original translation of the Bible was to Greek, the translation of the 70. And I think something went wrong with that translation because the original sin is always perceived to be the story of Eve giving the apple to Adam. And as we all know, it's usually men that give women gifts for various reasons. So it's probably the other way around, but something went wrong in that translation. But let's not go there. Anyway, original antigenic sin. What is the original antigenic sin? It's a big problem in infectious diseases, and it's a human problem because we live a long time, and we're exposed to agents all the time, and it's a particularly big problem in influenza because you get vaccinated against influenza, and you have pre-existing antibodies and a pre-existing immunity to influenza that was established somehow, and the vaccine doesn't work very well. So as a result, when you are vaccinated with influenza, the vaccine protects you poorly compared to what a good vaccine should be doing. Seasonal flu vaccine is actually a very important vaccine in many ways, but it's not the most effective vaccine. And for a number of reasons, there are up to 50,000 deaths from influenza in the United States. For certain age groups, it's one of the top 5 leading causes of death. Worldwide, millions of people die from influenza every year, and that's not during a pandemic year. So influenza is a problem because it's a highly diverse virus. And from a point of biochemistry, we can distinguish 3 groups of influenza: influenza group 1, group 2, and influenza B. Influenza B circulates only in humans. Group 1 and group 2 strains circulate in poultry and all birds, also in other species, and these are the species that usually cause pandemics. Now, to counter the effects of influenza, the vaccine until last year was a vaccine that comprised of 1 group 1 strain, 1 group 2 strain, and a B strain. And for a number of epidemiological reasons, now the vaccine is a tetravalent vaccine and comprises of 2 B strains that capture the 2 major lineages that circulate in the human population. Okay, so what happens is that when you're vaccinated, there is a good chance you may not get a protective response. In fact, last year, the wise people at CDC that looked at all the epidemiological data completely missed the strains to include the vaccine. And as a result, the protection by the vaccine was at best about 20%. A vaccine that's 20% effective is not a good vaccine. Many times they get it right, and so that's why it's worth being vaccinated. So immunity to influenza is completely mediated by antibodies. The antibodies bind to either one of two domains of the main protein on the surface of the influenza virus, which is called hemagglutinin, either the head domain, shown here, or the stem. Now, if you're one of the very fortunate individuals who, for whatever reason, produce super good antibodies that bind to the part of the virus that's in yellow, then you can be protected from pretty much most of influenza. It could be immune of influenza practically, or from most attacks. But that happens very rarely. Typically, those antibodies are difficult to elicit, and there is a worldwide effort, a major effort that's actually been now funded at a much higher level by the federal government and the military to develop a universal vaccine that you're vaccinated against once or maybe twice, and then you're protected for life and you don't have all the morbidity and cost and mortality that's associated with influenza infections. So, original antigenic scene. Vaccination with a seasonal influenza vaccine in 1 year may not elicit new antibodies. And you want new antibodies because you have a new vaccine. So you want a vaccine to elicit new antibodies compared to what antibodies are circulating. So how can we now use the tools that I described before to qualify this effect? Well, here's how we do this. We measure and determine the antibody repertoire, each of the antibodies that was present in the blood in 2013, and in this particular case in 2014 in our donor cohort. And in a young person, you see the development of a large number of antibodies that did not exist before vaccination. They were elicited by the vaccine. The vaccine did its work. However, if you're middle-aged, and middle-aged is defined in this case by being 45 and above, or elderly. Elderly is something that is forbidden in my group, but my students still use that term. It's called mature, okay? Then the vaccine does very little. There are very few antibodies that are elicited by the vaccine. Sure, you get more production of pre-existing antibodies, but not new antibodies, and that's a problem. Or is it? But this is true for all donors. This is a donor that's even more mature, about 77 years old. And in this case, the vaccine really does nothing. The reason why this is so is complex, but one way in which the pharmaceutical companies and the community is trying to mitigate that problem is giving people above a certain age a higher dose of vaccine in the hope that they will be protected. So, is it terrible? Does that mean that if you're above a certain age, your pre-existing immunity will basically doom you to be susceptible to influenza? It's not quite that simple. There is a silver lining. And the silver lining is as follows. If you look at people as they age, and this is the cohort that we studied, this is in collaboration with Sanofi Pasteur vaccines, what happens is, as you get older, you produce your antibodies in the serum, your repertoire is dominated by antibodies that bind to two different very kinds of flus. And that means these antibodies target specific areas in the flu virus, okay, that is highly conserved from year to year. Some of these antibodies do not really block the virus from entering the cell. However, these antibodies we found in a paper that we published a couple of years ago in Nature Medicine, they bind and actually they collaborate with other immune cells, if you're not immune compromised, to allow your immune system to recognize tagged virus. They don't block the virus, but they tag the virus so that the immune cells can come in, the macrophages and other cells, to actually wipe out the infected cells and the virus. So the bottom line is, as you get older, you're actually much more resistant to getting the flu, and this was well demonstrated in the 2009 pandemic, which actually hit younger people much worse than older people, okay? However, if you encounter a new strain of flu, which is very different from what you've experienced through your history, then you have a problem because your immune system cannot work well. So this is the kind of information that we're interested in. This work is being done by Jiwon Lee. I want to acknowledge Jiwon Lee for a number of reasons. He is a proud Cornell grad, worked with Madelisa for his master's degree, and he's been a terrific student who started his career at Dartmouth. And he, as I mentioned before, will take on the project and a lot of the flu studies that we're doing in the lab with him, and hopefully will help in a substantive way in the development of a universal vaccine, which absolutely is possible, but will take a long time to produce. So I would like to thank you for your attention. I managed to use my time. In fact, I managed to even go over. Just to remind you what I think Mike and everybody else in this room thinks, we will never be old. Old will be always 50 years older than we are. And I just want to again thank Mike for being the inspiration for my career, for your unyielding support for all these years, and for being a great friend.
So one thing, I want to make a note that with all of our speakers today, including George, not so fast, George, not so fast. We're going to, time permitting, allow for some questions. We have a lot of friends, but also a lot of younger students and postdoc trainees here, so we will have time for questions. But because we have a break scheduled from now and for about the next 20 minutes, I'd like to reserve questions for George for in the break time. So if you'd like to ask George any questions, you can come find him there. But let's thank him again for a really great talk. And so we'll join you all in the break area, and we'll reconvene here at 10:45. Thanks.